Welcome to the second of our videos on NIH grants, analyzing the overall structure of a grant application, specifically the significance, innovation, and research. So this is the second of a four-part video sequence on writing a research plan where we're using the 12-page NIH format. Here we're going to focus on the large-scale architecture of a document and compare two examples that won NIH funding. In this video and subsequent videos, we'll look at two winning grant applications and analyze them. And these are available from the NIAID website, and they are the Parrish and Ratner applications. There's the link to the NIAID site, and you can also find PDFs of these research plans at the CLIMB website. Note, to take full advantage of this and other videos in the sequence, you should have read the research plan of both applications before viewing. We're going to dive right into the technical details, and it's important for you to have some background in them in order to follow what we're trying to explain. So, let's look at the big structure of the Parrish application. And its subject is the structural and binding properties of the parvoviral capsid. And specifically, he was looking at two parvoviral capsids, one in a feline virus and one in a canine virus. So let's look at the big structure of this application, its significance section, its innovation section, and its approach section, and how this researcher organized this 12-page section of his proposal. First, in significance, he specifically highlighted the importance of this work, and he was extremely general. He said it's important to understand the fundamental aspects of virus infection processes controlled by receptor antibody binding, and the controls of viral host ranges. Then he explicitly stated the critical barriers. And the critical barrier here is, if you don't really understand how the parvoviral capsid works, you're not going to be able to advance antiviral therapy and vaccination approaches. And then finally, he focused specifically on improvement of scientific knowledge. Again, recall the NIH criteria in our initial video. It's critical to understand fundamental viral mechanisms to understand these parvoviral capsids and clarify the textbook knowledge of virus structures and functions. Next, on innovation, he pointed out that canine and feline viruses allow for the studying of a complete virus infection model, various ways the virus gets transmitted, the way it alters over time. And the methods being used in viral studies include combinations of kind of traditional methods, but the combinations themselves are novel. And then the heart of his section, the approach section. And he organized that by each specific aim. And his first aim was define the structural variations in parvovirus capsids, determine effects on capsid functions and the DNA release of that viral element into the cell. And we're looking here at the high-level organizational structure of this aim. There are substructures under C1a, C1b, C1c, but I think it's important to sort of look at the specific overall structure. First, he gives a background in this aim, and he gives a brief previous research review and the team's own preliminary results. Next, he talks about the studies of capsid structure that he is going to undertake. And then finally, outcomes, potential problems, and difficulties and alternative approaches. And recall how that meets the criteria of the approach section that we reviewed in the first video. The second aim was to define the structural interactions between the various parvovirus capsids and the variants of a specific receptor transferrin receptor or artificial receptors. And note the structure of this section, how it mirrors the first one. We have some background on receptor and capsid structures involved in binding. And that includes some literature review. Then there are a series of explicit or implicit research questions that fall under this aim. How does the capsid bind the TFR? And is that specific interaction important for infection of cells? And he talks about his technique of using cryo-EM and mutant mapping to obtain better structural models of the interactions between the feline and the canine viruses. And then finally, can the alternative receptor binding ligands in the CPV capsids mediate infection? And then what are the expected outcomes, possible problems, solutions, and alternative approaches? Again, he repeats that structure of looking at outcomes, potential problems, difficulties, and alternative approaches. And then finally, we have the third aim, using antibodies to probe the capsid structure and determine how binding to overlapping sites leads to neutralization in some cases, but not others. Again, same overall structure. What's the background? Antibody and TRF binding. Is the affinity of the antibodies important for the TFR competition and neutralization? And then finally, addresses expected outcomes, potential problems and solutions, and alternative approaches. So now you can see the overall large architecture of this parish application. He hits on importance, critical barriers, and improvement in scientific knowledge in the significance area. He addresses the nature of the novel methods that he's using. He provides background for each of his aims. And each of the aims are separate and unique. And the organizational structure is easy to follow. You see the big picture. Now let's look at the big structure of the Ratner application. This application aims to define the role of vaginal lysine, which is produced by the Gardnerella vaginalis pathogen. And this particular pathogen is a critical component of bacterial vaginosis. So again, we'll look at the significance, innovation, and approach sections of this 12-page section of the Ratner application. Now, Ratner takes a much different approach than Parrish in the discussion of significance. First, there's an opening context-setting paragraph. It implies the importance and improvement in knowledge. The importance is the size and the cost of bacterial vaginosis, and there are specific numbers associated with that size and cost in that section. And then the improvement in knowledge is the pathogenesis of bacterial vaginosis is simply not well understood. But our team has a novel approach to understanding that. Then there's a second paragraph which talks about the bacteria itself. And this is a previous research review. The next paragraph talks about the difficulty in establishing an animal model. And it's another research review talking about all of the work associated with trying to replicate the human pathogenesis in an animal environment and how that hasn't worked out well at all. And this leads to an overarching hypothesis that the virulence model of G. vaginalis is really human-specific. The next section talks about cholesterol-dependent cytolysins and a research review of the role of these toxins in pathogenesis in general in bacteria. The next paragraph focuses on vaginal lysin as a novel and human-specific characteristic of this particular pathogen. And it deals with their work in characterizing this CDC, and that it can use a particular receptor, CD59, in the pathogenic activity of the bacteria. The next paragraph talks about host immunity, cytokines, pathogenesis, and it's another research review, and it focuses on the role of interleukin-1 beta activating immune response, and it also focuses on their work in demonstrating the link between VLY and IL-1 beta. Now the next paragraph focuses on the CD59 receptor, and it explains how VLY co-opts CD59 and binds to CD59 as part of the pathogenesis. And then finally, the last paragraph talks about broadening the host range of vaginalis, and this gets to the heart of their research, where they're going to address strategies to alter both the bacteria and the host to address the main critical barrier of understanding bacterial vaginosis, and that is there's no animal model where we can replicate what happens in humans. Now, what's really interesting about this significance is it lays out methodically the logic of the overall application, where the research was, what the issues are, what the structural and functional components are, and what they're going to do in their research to vault that critical barrier of there's no animal model. They're going to make an animal model. Which that leads to innovation. And so the innovation here is they're going to genetically manipulate the microbe itself and then the host to create that new animal model. And they mention the innovative methods that they're going to be able to use to do so. And then we get to the approach section, and this is organized differently than the parish application as well. The first section talks about preliminary studies done by the team, what they've done so far, identify and characterize vaginal lysin, how they've manipulated the pathogen, their new marine models, how VLY binds to CD59 and mediates species selectivity, how it focuses, that's a very specific human interaction, and how HDC59 initiates various signaling functions that create the environment for inflammation and the biofilm formation by G. vaginalis. So it lays out all the previous work that they've done. Then there are two aims. The first aim is to define the determinants of the pathogen virulence using new genetic techniques, and then there are various sub-aims. And if you notice on sub-aim 1A, we've just listed the structure of that one aim. The first aim is 1A, determine genes required for production and regulation of VLY. And then there's experimental design, anticipated results and interpretation, and potential pitfalls, alternative approaches, and future directions. And that particular sectional structure gets repeated in each of the sub-aims. And so aim 2 is designed to determine the role of VLY and G. vaginalis at the host pathogen interface in vitro and in vivo. And now you can see the overall structure of this Ratner application. A lot more emphasis, a lot more text was in the significance section than in the parish application. But it was really important for them to do that, to set the logic of what they are trying to accomplish in this research proposal. So let's compare the two. Parish is on the left, and Ratner's on the right. Notice the difference in the way the applications are structured. Now, each approach section deals with the specific aims, but the organization is slightly different. In the case of Parish on the left, significance is a more general discussion. In the case of Ratner, significance really lays out the logic of the research, where the research was, and where we're going. And the approach section in Ratner really focuses in on the experimental design, each specific sub-aim, and where things are going. So the approach section in Parish is much longer than the approach section in Ratner, because it was important for Ratner to lay out the significance logic in that section. So step back, take a look. There's no right way to organize your 12-page significance and innovation approach section. Each research project is different. Each research project has its specific challenges. But as you can see, there are commonalities here. We look at the importance and significance of the research project. What are the critical barriers associated with it? What are the innovations? And then what are each of the aims? And what are the experimental activities within each of the aims? Those commonalities exist between applications.